It's Johnny again. I'm back at NeurIPS at the poster sessions from the Polaris team. I was walking around and I saw MassSpecGym, which was one of the most recent certified data sets on Polaris. So I'm here with Roman, who's one of the, I think the first author on the paper. Do you want to quickly start by introducing yourself?
Yeah, sure. So my name is Roman. I'm a second-year PhD student based in Prague. In the groups of Tomasz Pluskal and Józef Siwiec. My background is mainly computational, but I've been working in computational biology for like 3 years.
Amazing. So for those who aren't familiar, do you want to quickly just describe like mass spec, why would someone care about mass spec and what that means for maybe drug discovery applications or molecular discovery?
Yeah, sure. So basically the motivation is super simple. If we take like a human blood sample or any random plant and ask a question, how many molecules have not yet been discovered in there, like the answer would be quite pessimistic. So basically in human or in plant kingdom, we know only less than 10% of molecules. And with our benchmark, we just want to abstract away this scientific discovery of new molecules into a machine learning problem, so that people can contribute new machine learning models and see how well they can discover new molecules from nature.
Cool.
And mass spec actually is the most prominent technique which allow us to sort of abstract away this process.
Cool. And so what are some of the challenges that you're seeing in terms of the data sets and the benchmarks available in people who are doing work in mass spec?
Yeah, actually, probably the main challenge is just to somehow unify the opinion of many people. So in our project, we have like a community-wide effort, but people have been working on these problems for like 20 years, so everyone has their own opinion. So it was kind of challenging to standardize all the opinions, but yeah, we have some benchmark now. I think everyone is more or less satisfied. But of course, like it's probably in computational biology in general, it's hard to collect new data.
Yeah, cool. So tell us a little bit more about sort of the work that went behind the new data set, the curation, and maybe a little bit of the benchmark that you proposed on top of this.
Yeah, sure.
Yeah.
So as I mentioned, our benchmark is about discovering new molecules. Everything is defined in terms of pairs of mass spectra and molecular structures. And our benchmark has three main components, which are shown here. So first component is that we define three machine learning challenges. One is like the ultimate kind of problem of mass spectrometry, given a mass spectrum to decode the molecular structure. And this is kind of similar to the goal of AlphaFold, which predicts the structure of a big molecule. But here we're interested in the structures of small molecules. The second problem is the simplified version of this first problem. Instead of predicting a new structure from scratch, the goal is just to pick the best molecule from some reference candidate list. The third problem is an inverse one, to simulate a mass spectrum given a structure. So this is the first component of the benchmark. The second component is that the largest publicly available data set of mass spectra labeled with molecules. We try to do our best to really make it high quality and well standardized with all the metadata ready for machine learning. And the third component is that we have a generalization demanding data split. What it means is that if some model has really good test performance, we just want to make sure that it's not because it sort of memorized the train set, but it's because it really generalizes to new data in test set.
Cool, amazing. So you can find more about the data set and the benchmark, obviously in the paper. We'll have a link down in the description, but you can also access the data set on Polaris. Thanks again for the great overview of the work. Awesome.